Hello, everyone. Welcome to the podcast for day 17. In this podcast, we're going to cover objectives 1 and 3. It's really difficult to kind of separate these into two separate ones because they're critically linked. Now, as I recommended, you should have watched Objective 2's podcast first. That was the one on mutations. And in this podcast, we're going to deal with the cell cycle and checkpoints and how, when the cell cycle fails, we can allow mutations to occur and to persist within a cluster of cells. So let's go ahead and get started. To get us started, let's go ahead and write down the cell cycle. This is the same cell cycle we learned a few chapters ago, or a few days ago. And so let's go and write cell cycle up here. And if you remember, we started off with a cell here that just came from the previous cell cycle, from the previous M phase. Remember, this also produced another cell too that went off and began its own cell cycle someplace else. But this cell here entered into G1. And then from there, it goes into the S phase. And then from the S phase, it goes into G2. And then from G2, it goes back into M phase. And then we'll begin the cycle all again. Now, if you remember when we went through this cell, the cell in G1 got a little bit bigger. In S, it copied the DNA. In G2, once again, it got a little bit bigger. And then in M phase, it segregated the chromosomes. As we did previously, we drew chromosomes within these cells as we explained it. And let's go ahead and do that again, but we're only going to draw one pair. So this might be like chromosome 1. And we're going to draw the centromere here in the middle, just like so. We also have it in this cell, so we'll put it here just for good measure, but we're not going to follow this cell anymore. It's off to do its own thing, and we'll never see it again. Sad day for that cell. All right, in G1, we still have our 2 chromosomes in the nucleus. The cell gets bigger. Remember, we said at this point here the cell is getting bigger. It's checking for mutations. It's making sure all the enzymes are present to enter into S phase. And in an S phase, these chromosomes copy themselves. So this should all be review, but it's a good review to have right now. So remember that the chromosome here I wrote on the left is equivalent to this one here. And the one that's kind of curving from it, that's our new chromatin that just got copied. And same thing with this other one. And we still have them over here. So let's go and just draw them just for good measure. And then in M phase, just to remind you, let's go ahead and where should we draw this at? We'll draw it up here. So let's draw our M phase cell here. I'm going to draw a little bit bigger just because it's easier to draw the things inside of it. Remember, in the cell cycle during M phase, let's just focus on M phase right now. We had our centrosomes forming here on either side. And they would shoot out spindle fibers, microtubules. So let's just go ahead and have some microtubules coming out from the centrosomes. And remember, these attach to the chromosomes. So let's draw our chromosomes now. And in this way, remember, these green microtubules are going to constrict and pull the chromosomes over into this cell. And same here. And then at the end of this, what we'll end up with is our 2 cells such as we see here, right? So this will go like so. All right, this should all be review for us. But let's introduce some new concepts here. The first one is, well, let's present something else here real quick. For this to occur and to happen where we produce 2 cells, and remember, the goal of mitosis is to produce 2 identical cells. So in order to do that, the cell has to pass through what we call a cell cycle checkpoints. Your book refers it to as the cell cycle control system. And so we have one here, we call it the G1 checkpoint. I'm just going to write G1 check. At the G1 check, it will do is it will hold this cell in check. It won't allow it to move. In fact, it holds it in a certain state called G0 until it's past this checkpoint. So during the G1 check, it's checking for a few things. One, it's checking to make sure there's no mutations. We don't want, well, let's just write checks for mutations. It also checks to see if the cell has the correct enzymes made and that are necessary for the next stage of the S phase. It also will check to make sure the cell is the correct size. So we'll go check size. But really the first two are the most important things here. Once it's checked this, verify there are no mutations and the enzymes are present, it will now go into S phase. And it will copy the DNA through replication. There's no checkpoint in S phase. The next checkpoint comes with the G2 check. At the G2 check, it makes sure that all the DNA is copied. We'll write DNA copied. It'll check that there are no mutations. And these are the big things. But it also checks to make sure the enzymes are ready and it's prepared to go into M phase. Then our last checkpoint occurs during mitosis. More specifically, it occurs during metaphase, sort of as we see here. So I'm going to put the M checkpoint here. And what we're checking here, there's really one main thing we're checking, and that is that every chromosome is attached to a spindle. So I'm just going to write spindle check here. It makes sure that each of these chromosomes is attached to the centromere with the spindle fiber. This is really important because if it's not attached, then we cannot properly pull these chromosomes into the new cells. So these are our checkpoints. And as long as these are working, nothing's going to happen. Our cells are going to keep dividing, they're going to keep growing, and they're going to keep producing identical cells with no mutations in them. This is going to make this slide a little crowded, but I think it's important that we keep this figure going here. And there are two broad classes of genes we need to talk about. They are tumor suppressor genes. So tumor suppressor genes. Tumor suppressor genes. And we have another class of genes we need to talk about. And these are called Proto Oncogenes. Proto Oncogenes. Now, what do these genes do? Well, they're cell cycle genes. They're involved in regulating the cell cycle. And the tumor suppressor genes, I like to think of these as the brakes of the cell cycle. So I'm just going to write stops. Okay, they're the stop sign of the cell cycle. They're the brakes. Meaning, as you're moving through the cell cycle at the G1 checkpoint, if the cell detects mutations or if it detects the enzymes aren't ready to copy the DNA or if the cell's too small, these enzymes, the genes that then encode the enzymes, will stop the cell cycle right here. So the tumor suppressor genes stop the cell cycle here if there are any mutations or if it's just simply not ready to move into S phase. Likewise, in G2, there are other sets of tumor suppressor genes that check if the DNA has been copied and whether or not there are mutations. If it detects mutations or if it detects that all the DNA hasn't been copied yet, the tumor suppressor genes stop the cell cycle. That's their job. All right, then once again in M phase, if it detects that the chromosomes have not attached to the spindle fibers during metaphase, the tumor suppressor genes will stop the cell cycle. All right, so what do the Proto Oncogenes do? Well, since I grew them in, I put them in green to signify that they're the gas pedal of the cell cycle. So I'm just going to put go, not to be confused with geo. Maybe I'm going to put start, because I don't, as you're looking at your notes later, I don't want you to mistake that for this GO over here, which was where the cells hang out until they're ready to move on. So let's just put start. So they start the cell cycle, or they allow the cell cycle to proceed forward. So as we're moving along here, we hit G1, all the mutation, there's no mutations. Well, let's back up. It stops here because of tumor suppressor genes. Make sure there's no mutations, and make sure that the enzymes are present. Make sure it's the correct size. Once that's been determined, tumor suppressors stop doing their job, and now the Proto Oncogenes, they apply the gas pedal, and now the cell moves into S phase. Okay? In S phase, they copy their DNA. They then move into G2 phase. At G2 phase, the tumor suppressor gene stops the cell cycles, checks to make sure the DNA is copied and there are no mutations. Once that is verified, the proto oncogenes hit the gas pedal and move into M phase. Once again, the cell is stopped, the cycle is stopped by the tumor suppressor genes to make sure all the chromosomes are attached to the spindles. Once that's been verified, tumor suppressor genes stop doing their job, and now the proto oncogenes apply the gas and they proceed through mitosis and complete the cell cycle. So as long as these two genes are working, everything's good. Now, as their names apply, tumor suppressor and proto oncogene, these genes, when mutated, can lead to cancer. Now, how do they lead to cancer? Let's just look at tumor suppressor genes first. Let's pretend our tumor suppressor genes, and we have several tumor suppressor genes in all of our cells. They're good genes. They're supposed to be there. They help us from getting cancer. But if one of them is mutated, what can happen? Well, actually, let's put down a couple of examples of tumor suppressor genes. A classic one is P53. And another important one that you'll certainly hear about at some point, maybe frequently, is BRCA1, BRCA1 and 2. These are tumor suppressor genes. BRCA1 is linked to cell cancer. P53 is linked to almost every cancer. If these are mutated, now when the cell comes to G1 and wants to check to see if there are mutations, but it can't stop, it's going to fly right by that stop sign, even if there is a mutation. Now, if there is a mutation, what happens now? We just copied that mutation in S phase. Over here, we come here. If there is a mutation in the tumor suppressor gene, once again, it can't check for mutation, and we will fly through that stop sign and move into M phase. Once we're in M phase, we now have mutations. Why don't we do this real quick? Let's just make a black dot here and pretend that's our mutation. And what we have done is we have copied that mutation on this cell here, on that chromosome, and we've recopied it over here, right? And now over here we still have that mutation, and here's just shown in black. And now after M phase, we have just created a new cell that has this mutation. Two new cells each have the mutations, and that's because the tumor suppressor genes blew through the stop signs and allowed that mutation to persist within this cell. As you keep doing this over and over and over again, you're eventually going to accumulate a lot of mutations. And at some point, the cell will start behaving like it's supposed to be. Like if this was, say, a cell in your pancreas. At some point, it's not going to stop being a pancreatic cell and start being a cancer cell. We'll talk a little bit on probably the next slide, about what happens when a cell becomes a cancer cell. But let's finish our discussion of these two classes of genes. All right, now, what about proto oncogenes? Well, proto oncogenes are a little different. What happens when they get mutated and they cause a problem? They now are called oncogenes. Okay. They get mutated in an interesting way. Instead of being mutated where they don't function anymore, they're mutated where they are overexpressed. Proto oncogenes that become overexpressed. So, for instance, if normally a proto oncogene is expressed at ×2, just to make a number, then oncogenes might be expressed at ×10. They're expressed at a much higher level. And when I say expressed, what I mean is they are transcribed, and then they are translated, and they are now active proteins. These active oncogenes now, because they're overexpressed, we call them oncogenes now. It's kind of like if you're driving a car and you push your gas pedal all the way to the floor and you start driving super, super fast, right? And that's what's happening here. When the proto-oncogenes become oncogenes, they fly through the stop signs, even though the tumor suppressor genes are working, but they refuse to stop. They keep moving. And in the exact same way that we saw the problem with tumor suppressor genes, the proto-oncogenes becoming oncogenes go through these stop signs, ignore them, allowing these mutations, similar to these ones in black, to continue forward. In this way, we accumulate mutations. And these mutations, if they occur in the appropriate numbers, then they can lead to cancer. Okay, now this ends our second podcast for this chapter. There's one more podcast that will talk more specifically about cancer, what makes a cancer cell different than a healthy cell. So make sure you tune in to watch that one as well. If you have any questions, please make sure you contact one of us instructors and we'll be happy to help you. We'll see you in class. Bye.